here we are. Whoops. And here we are. Back in class. And I still have to grade the drawing assignment we did. The students that turned it in. Some students sent me pictures. I've graded those. I just need to get to the ones that were the physical ones, and I'll get those back to you. And that's where we're at with that. Then, reminder that, remember, you have to participate in our course. So that means that either sign the sign-up sheet each time, we have to try to get here in the 20% course. So that's described in the syllabus, and I think everybody's familiar with it. But I just kind of remind you, because I think it's probably the only class you've had that's like that. And then if you miss, within 1 week, you have to send me notes based on the recording. And I think the recordings are going well. You know, sometimes there's a little technical issue, but they seem to be going well. And so we're doing that. And that's the rule is you have to participate, you have to keep up with it, like that, okay? So that is where we're at. And then I met with Riley yesterday, and I think, you know, his first visit here was great. Riley's going to visit 3 times, and he's going to visit again in like late March, like March 24th, I think, and after our spring break and after our first exam. And just to tell you, when he comes back, the second visit, he's going to focus on the receptor, this CAR receptor, this chimeric antigen receptor, this modified receptor, and how it got built, and how it functions in the cell, and then what the signal transduction pathway is. And so that's a chapter 17 topic for us. And so by that time, we'll be extra experts on that. But we already know some about it. For example, one of the things that the CAR does, it activates the RAS, MEK pathway, because it's going to stimulate proliferation of those cells because they want more cells in the patient, and they want the patient's cells to persist. They want to fight cancer for a while, like that, okay? So Riley talked about the receptor the second time and the signal transduction pathways. Then the third time, it's going to be about the cellular response, because that's always what's happening. We've got a target cell, and ligand binds the target cell receptor, and it activates the signal transduction, and there's a cellular response. And the cellular response is proliferation, but there are other things that this T cell is doing, so he's going to talk about that. And then later in our course, he's going to come in and talk about chapter 20 topics where it's actually talked about in our book, the CAR T cell therapy system, and more about sort of the actual, patient strategy of how we get this done and how it might look in the future. Right now, it's prohibitively expensive. It takes a lot of stuff and a lot of expertise. It's only done in a few places. But, the analogy is like the first time an airplane was flown. It's like, okay, I wouldn't get on that, right? It's still experimental, but there's a lot of success, and the success is driving a lot of funding from the government and a lot of funding from private companies. So that technology is going to jump ahead, similar to how computers and other technologies like that, right? So it's going to get cheaper, it's going to get streamlined, it's going to be done in more places, and it's going to be done for other applications, not just cancer and so forth. So it's exciting. It's just a good thing for us to think of Riley. But also remember, Riley is a tutor, able to talk about the content, and he's really good at that. We saw he's a good speaker, but he's really good in just chatting like that. And he said nobody's contacted him yet. That's fine. We haven't had an exam yet. That's what he said. But this is ongoing learning. So, you can contact me with questions. Students are doing that. That's great. But also you can connect with Riley. And that's part of what is, he really would like to help like that. He's enthusiastic about these topics, loves to talk about them. So, if you want to talk about them a little bit, you can talk about it with me. And a lot of times when students meet with me, they're kind of apologetic. Well, you don't need to be, and you also don't need to be apologetic with Riley. He and I love to talk about it. Like, oh, yay, right? I always say it's like, when you ask somebody else, like, who likes sports, you're like, hey, who's your favorite team? And it's just like, whoosh, like that. So it's like that. So don't hesitate to contact Riley. Then this week, I'm going to start advertising for next school year these biomedical research internships that take place at Maine Health Institute for Research in Scarborough. And they're the best. And we have students doing them right now. We have about a dozen students down there each year. And it goes from September to May. And if you like these topics, that's what they do. It's all molecular genetics. It's all this stuff. And it's all using mice as a model system because of the orthologs in mice and how similar they are to us, in their molecular mechanisms. And it's about cancer biology, it's about stem cell biology, it's about heart and especially circulatory systems. So blood vessels, they're world experts on blood vessels down there and have been for years. It's also about bones. They're world experts in bones and bone development, bone health. And another big topic they have is fats. There are some different types of fats in our body, and they have really, so not just white fat that is part of obesity, but brown fat and tan fat and so forth, like that. And they're becoming world experts on that too. So it's just super exciting, and it's all this. It's just stunning. Like molecular medicine is just all this kind of stuff. So just so you know, I'm the person to talk to about that. And it's just really it's significant. It's a really good opportunity. Okay, if you're going to be around. Okay, so today we're starting to talk about chapter 9, and so the first thing is, well, we know a lot about transcription right now because it's claim out of video, and that's what this chapter is about. And molecular biologists love transcription factors, so we're going to add some to our transcription factor knowledge in this. And this is a correct, good, valid model like that, and we're going to think about it a little bit differently because we're going to think about more about the actual structure inside of the cell and the proteins that are bound. And there are lots of proteins that bind to the promoter, and that's one way that a protein like MyoD can help the rate limiting step and get myogenic transcribed, is to help get those proteins bound to the promoter, get RNA polymerase there in place, and it's called the preinitiation complex, like that. Then another way we're going to learn about is some other transcription factors actually have to unfold the DNA. The DNA is actually coiled up in around proteins called nucleosomes and structures that are overall called chromatin. So we're going to remodel the chromatin because sometimes that's the rate limiting step. The preinitiation complex could assemble fine, but we need to unfold the chromatin, just like I said, like unfolding a blanket because you're going to use it on your bed. And then the third one is the surprising one. That's the person that won't let go of the hamburger on the plate, and is just talking with you about everything under the sun. And there is the rate limiting step at the fair, is that person. And that happens here where it's kind of the last step. The preinitiation complex is fine, RNA polymerase is landed, the chromatin's unfolded, and the RNA polymerase takes off, starts going. It goes about 70 nucleotides, and then it just stops. And it arrests or pauses, that's what they're called, and it's a transcription elongation. And Joe pointed out, wow, there's a lot of detailed words about elongation in the book. For us, it's just arrested polymerase, and let's learn about that. Let's introduce it like that. And that's the rate limiting step. So we'll learn about transcription factor that regulates that. So those are our big 3 things for that. And that's a big part of the chapter. Before we dive into it, I just want to loop back over a little bit of what we talked about because it's so important. And that is, when we think about oncogenic retroviruses, we're going to think about a few of them. And for our first exam, we'll talk about RAS and Mick. And we just want to emphasize that they're part of a normal response, right? So a normal response, like this kind of response, and that's the PDGF receptor and activating RAS, RAF, MEK, ERK, like that, ELK, under a scab, right? And then that pathway, we'll learn about in chapter 17, that's used by a lot of different receptor tyrosine kinase receptors, like that. So it's a common pathway, and it could get, some components of the pathway could get mutations, and that could lead to cancer. And we learned that in Mick, the story is, when it contributes to cancer, is the problems are in the regulatory region. So Mick is highly carefully regulated, and what happens in cancer is that it gets connected to a regulatory region that's really strong, and you just make a lot of Mick, and it just overwhelms it. And so last time we said Mick is a transcription factor, so we want to go back to this list, and we'll keep building this list throughout our course. And I just bundled these all together. Mick is just regulated by a whole bunch of different ways, like that. So I just put them all together. And there are other transcription factors that are regulated by one or the other of these, but very few that are regulated in all these ways. And one of the reasons we say that is to underscore how dangerous Mick is. When Mick is active, it binds to genes that drive the cell through the cell cycle and proliferate, like that. And so we said, so when we think about Mick in cancer, we think about 3 examples. These 3 we talked about last time, and all of them involve changing something about the amount of Mick that's produced. The first 2 are let's give it a different regulatory region. Either a retroviral regulatory region that's just going to make lots of Myck, or a chromosomal translocation. Those are two very different stories like that, but they have the same effect. Too much Myck. And the third one is this kind of curiosity, where you just accidentally duplicate the gene a bunch of times. So you just have many, many copies of it, and you make too much Myck that way. So we talked about those, and it just, here's the way they write about it. It says, generally, Myck overexpression does not stem from point mutations in the gene. In general, there are these other mechanisms, right? And in intro bio, you kind of learned, oh, mutations, you could have point mutations, you know, insertion, deletions, and so forth like that. But there are these other chromosomal rearrangements, like a translocation or a duplication, that can be problematic. So that's what they're trying to say there. And we want to just really emphasize that Myck's story in cancer is different than RAS's story, right? RAS is about mutations in the coding region, point mutations that alter the amino acids in the RAS. That's what's happening. And that should make lots of sense. And the reason I'm going back over this is stuff that I would say, oh, I would kind of hope that you would just, I will never forget that. You know, like when I'm hanging out, you know, sometime on the beach, I'll just be like, yeah, just thinking about RAS, you know, what a rowdy protein that RAS is. And then Myck, and what a rowdy Myck protein that is, you know. And they're in our bodies right now doing good jobs, you know, carefully regulated, and they could get out of control. So we have a really good compare and contrast with Myck and RAS. And scientists have known about them since the 1970s because they were found in oncogenic retroviruses, and we know how and why. And then they've just been studied like that. Okay? So great. So any questions about that before we roll further into chapter 9? So the book talks about this. The book doesn't really express this list because it's just organized differently. But this is a really nice list. And the way we started talking about it, yes, Godwin.
This is, so you want us to add the CAR-T lecture to the chapters that we're going to study for the first exam?
That's a good question. And the stuff that we've learned about CAR-T so far, it's just in class. It's just what Riley has told us. And there could be a question or two on the first exam about what Riley told us. The first exam is on March 9th, so we're getting to it like 2 or 3 weeks away from it, Thursday, March 9th, like that. Yep. But just a little bit of CAR-T. And in our textbook, CAR-T is just in one figure in chapter 20. And so it'll really be mostly what Riley teaches us that will be on our second exam and the final exam. Like that. Yep. Okay. Other questions people have about anything at all? All right. So let me just say, in chapter 9, before we talk about these examples, chapter 9 starts out with a couple sections on regulation of transcription in bacteria. And that's good because there are our very, very distant related ancestors. And we don't want to learn about that because it's microbiology. You could take microbiology. What we just want to learn about it is what I've already said, and that is that our clay model video is a universal model for gene organization. There are 2 parts in bacteria also. They use different names for them, and that's just too bad for student learning. It's just sad. But genes always have a transcription unit that is going to be the output, the functional product of the gene, and that's why it's called a gene, because it has a unit of output and then a regulatory region. And bacteria have that too. Bacteria regulate gene expression. They don't make enzymes for certain nutrients unless the nutrient is there. And then it's like, let's make some of these enzymes and get that nutrient eaten up. So you can just breeze through that. It's required reading, but that's why it's there, like that. Then another thing before we dive into it is that we're using good terms, right? So people float around with these terms a little bit, but just look at this statement. And we're like, yeah, we're nodding to this. Enhancers are cis-regulatory elements in the genome that cooperate with promoters to control target gene transcription. Unlike promoters, enhancers are not necessarily adjacent to the target gene and can exert their functions regardless of enhancer orientation. You could turn it around, positions, you could move it further, you could move it downstream, and spatial segregations from target genes. I don't know what that last one means just off the top of my head, but that's how we drew it. And that's just a nice way. And that's people like us writing about it, explaining it. And so those are good terms like that. And you'll just, in some other courses, you'll run into a little bit different use of the terms, but the concept is the same. And it's just really important for us to appreciate it because this is the centerpiece of molecular biology. Another thing I want to mention is that we didn't read Chapter 8, and Chapter 8 just brings up this, that in Chapter 9, we're especially going to be talking about coding genes, and coding genes are synthesized by the RNA polymerase II. In our bodies, there are 3 RNA polymerase, 1, 2, 3, and all of the coding genes are transcribed by RNA polymerase II. So I just want to say that once, because once in a while, our book will say RNA polymerase II, like that, and it's just saying that. And you can see these are non-coding genes, and RNA polymerase 1 and 3 are involved in non-coding gene expression. No big deal, we didn't read that chapter. Just pointing that out, okay? So you see we skipped some chapters, right? And that's fine, that's fine. All right? All right, so then here's the last thing I want to say before we start diving into it, is what we're going to talk about now, we're going to just package. We're going to roll through it. I'll just skip this slide for a second, and I'll show you this. We're going to talk about these 3 examples, and we're just going to pick one transcription factor that's a really good story for that type of rate-limiting step. MyoD, we know that. How does MyoD work? It does some other things, but the main thing that it does is it seems to help assemble the preinitiation complex. So we'll talk about that briefly. Great example. Do other transcription factors? Yeah, most transcription factors fit into these 3 categories. And there are 2,000 transcription factors in our human bodies. So most of them fit into these 3 categories. So there are lots of transcription factors that regulate the preinitiation complex in a lot of different ways. And we're just going to learn, MyoD. You want to talk about the preinitiation complex as a rate-limiting step? I want to talk about MyoD. Then for remodeling chromatin, we're going to learn about that. The book talks about remodeling chromatin a lot. And it talks about a lot of different types of stories. We're going to learn one. We're going to drive through it learning one, and that's what estrogen receptor does. And we're going to learn about estrogen receptor because it's a very, very famous transcription factor. Testosterone receptor is the same kind of story. So in all vertebrates, so not just us, but all mammals and reptiles and amphibians and fish, those are the sex-determining hormones in the body. So female vertebrates have estrogen, and estrogen receptors plays a big role. Males have testosterone receptor plays a big role, like that, okay? So we're just going to pick on estrogen receptor. And then releasing this arrested polymerase example, I mentioned before, most genes have that as a rate-limiting step. So many transcription factors regulate arrested polymerase. And we're going to learn about one. It's called heat shock transcription factor, or it's abbreviated HSF, okay? So we're going to just roar into this, and because it turns out it's a big field like that, of course, right? And our book kind of dives into it some, but we're going to hang in there with our clay model, and then these additions like that, and then there's another piece for Chapter 9 that we'll get to, like that, okay? So in doing that, I just want to say that there's some real surprising mysteries in this field, right? So we're going to lay it out like, a ton is known and it's making sense and it's physical. There are some remaining crazy mysteries. And so here's just one, and it's not a test question one, but just say, oh, wow, is there a reason to study biology? Yeah, because there's a lot of stuff that's just really unknown. And here's one. So a surprising discovery 10 years ago, 15 years ago, like that, is that we say, here's the enhancer, here's the transcription factor, and it's going to bend, the DNA is going to bend, and we're going to get it, to that preinitiation complex and RNA polymerase, and it's going to do its job there, like that, right? And so the enhancer could be way over here, but it's tethered, and so it's like on a leash, and it can get, bumped there, right? So this can be way over here, and it's supposed to be helping RNA polymerase here. And what these scientists discovered is way over here, in a seemingly useless situation, the enhancer and the transcription factor are getting RNA polymerases bound there and transcribing there, but just transcribing just junk. What's up with that? Like that. And people are like, and they call that eRNA, enhancer RNA, and they're like, is that up? And it's just completely mysterious. Like, what is that doing? Why is that doing it like that? Seems like just useless effort, and we don't see a lot of that in biology. Natural selection tries to make things efficient, so it just seems goofy because all the enhancers that are active are doing that. They have a transcription factor that's saying, hey, how about we start right here, right like that, and you're like, where's the preinitiation complex? All that stuff. It's just like, oh, just getting it done, trying to do my best. I'm a little tired, but yeah. So there are lots of advanced topics where you're like, wow, there's just like that. People don't know. The world experts are just like, seems cool and weird. Okay, so here we go with this, and we're just going to roll through these stories, right? So our first example is MyoD, and we're MyoD experts. Where is MyoD by? The enhancer of the myogenic gene. And it has other target genes as well, but our favorite is the myogenic gene, right? And not just an enhancer, remember the first quiz, not just enhancers, but that enhancer like that, right? So it binds, and then it helps it get going. How does it work? And the way it works is it activates the preinitiation complex. And there's one protein that it helps get the job done, okay? And so here's what this preinitiation complex looks like. Again, it's like chapter 8, but we didn't read that, so we don't need to know these details. But there's just a bunch of different colored proteins that are bound here. And this is right at the start of transcription. So there's the promoter, and there are these transcription factors bound to the promoter, and they're helping get the RNA polymerase there. And the RNA polymerase comes along with about 13 proteins along with it. So it's a great big bunch of proteins that assemble here that's called the preinitiation complex. And they all got to get put together. It's like a jigsaw puzzle, and you got to get them all put together. And so lots of times that's the rate limiting step. And lots of times it's one factor, okay? And that's what it's like with MyoD. And so we're just going to look at this, and it says the myogenic determination factor, MyoD, activates transcription of muscle-specific genes. We know that term, right? Muscle-specific genes, tissue-specific genes. It's going to help differentiate, and that involves diseases. By binding to consensus DNA sites, the enhancer found in regulatory sequences of these genes. Great, okay? All right, so it's going to help this one protein called TF2D. Or let's call it by that parentheses name. It's going to help a preinitiation complex protein called TBP. And so I'll just write over here that, because it's just a very memorable name for it. So here's the DNA for myogenic, right? And so there must be an enhancer out here, and our hero, MyoD, is binding there, like that. And its function is going to be to help get proteins bound to the promoter. Here. And there are some different ones, and I won't color in all these different colors, but not that color probably. But there's a purple one, and a brown one, and a yellow one. And some of these transcription factors, so those are just other transcription factors that bind to the promoter, and their function is they help RNA polymerase get right to the start. The promoter is always right next to the transcription unit because that's its function, is it's just get that, there it is, like that. It's like if RNA polymerase was an airplane landing at the airport, the promoter is just right at the start of the runway, and there are just little lights and signs going, this is it, this is the place to land. Not before that, or not after that, just right there. That's all they do. So those proteins are there. Some of these promoters include, right, these promoters, they're just letters, right? It's like A, G, C, T, G, 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 like letters like that. And some of these proteins bind directly to the promoter letters. and others bind, like that yellow one, bind to some other proteins, like that, okay? So these are letters, and what scientists found is that lots of promoters include a sequence that looks like this, TATAAA. So it's just a sequence, and they call that the TATA box, because it has those kind of letters, right? Does that make sense? It's just part of the promoter DNA, and some promoters have a TATA box. And who binds there? The protein we want to learn about. And it binds right there to those letters, and it's called TBP. So that's a great name for a protein, unforgettable name, because it's called TATA binding protein. And you're like, yeah, thank you. College students, thank you, scientists. So it's TBP, right? And what we would see, right, what we would see for the myogenic gene is that when the gene is off, before transcription starts, lots of these proteins are already bound. But TBP is not bound. TBP is there, but it's just not bound yet. And so MyoD's function is to actually somehow bind to the TATA binding protein, TBP, and get it there. And we talked about how, the long length of DNA is like a leash, and so the MyoD is leashed there or tethered there, and it can help somehow interact, right, like that. Yep?
Did you say that the TATA box is in a promoter?
Yep. Yes. So there are promoters, and the promoters are just made of DNA sequence, right? Like that. And a bunch of promoters have a TATA sequence in them. Like GGCAT and then TATAAA, like that.
And then you said MyoD tries to get the
Yeah. Yep. Yep. Yeah. So this is our example of a transcription factor that helps assemble the preinitiation complex. And specifically, it's helping with the TBP, TATA binding protein. Okay? Like that. That's it. We're almost done talking about it. And we could come back and make a clay model video and show that. The DNA would be bent, and MyoD would be a purple piece of clay, and it would be binding to a red piece of clay, and it would be helping get it there, get it assembled, okay? Like that, that's it. So that's our first example, that's the rate learning step. It's a lot of different complicated things, these are like cheeseburgers, right, with a lot of different toppings. And one of the toppings is TBP. Got to get that on there. It's not on there yet. Everything else is on there. We're waiting for the TBP, okay? So that's our first example. And it's, like I said, we could, make that, right? We could add that and understand it like that, okay? And so then just to mention one more thing is that I said, yeah, that's a good, then these stories get kind of surprisingly complicated because, just to mention this one more time and then we'll be done mentioning this idea that these stories can be complicated. So here's this, right? The myogenic MyoD protein works by interacting with TBP, okay? And then down here, MyoD is involved in 2 steps of preinitiation. First, TBP somehow stabilizes MyoD binding to the enhancer, right? So it's just like, what? We thought, the MyoD would just be there. But somehow this is like a 2-way street, where the first thing is, MyoD is just like, I'm not sure I can find the enhancer. And TBP actually binds to it and says, help, here's the help you need, there's an enhancer, like that. So it's surprising because it's a little 2-way thing. So first, TBP helps MyoD get to the enhancer, and then at a later stage, MyoD facilitates the association of TBP with the preinitiation complex, right? Just a little add-on, but it's just like, right?
It's like the 3 blind mice helping each other.
Yeah, it's a team effort, And so the way I drew it, right, is just a little bit more complicated, where MyoD isn't even quite bound yet, but TBP binds to it and helps it get there. And then now it's there, and it's this complex of MyoD and TBP, and now the leash is flopping around, and then TBP bumps into its site, right? So very physical and logical, just a little bit complicated, a little bit surprising, like that. You know, but cool. It's cool stuff. It's just like party down cool stuff. When you go to a bar, about half the people in bars are people who study transcription. Every bar, all the time. They don't talk about it because they're not, you know, hey, I'm a weirdo, but yeah. You really start pressing them. You're like, what's some of your favorite things? They're like, oh, TBP. And you're like, PBR? No, TBP. Yeah, okay? So that's it. We're done talking about MyoD and the Preinitiation complex. There are many stories about it at every bar in the world. Okay? So now we're going to go on to talk about the remodeling chromatin. And our book talks about that a lot. Because we're going to have to introduce what is that about. And then we're going to use our example, the estrogen receptor, to learn about it, okay? All right, so in doing that, let me just start with this slide and say that here, that's a new transcription factor for us. And then next one, that's a new transcription factor for us. So I just want to show that we're going to add these to our list. So, estrogen receptor, it's dynamic. It's a transcription factor that's dynamic. How is it dynamic? How does it switch genes on? And the answer is a hormone. Small molecules that bind to proteins are often called ligands, just a general word. And so estrogen is a ligand for the estrogen receptor. So we want it on our list because it's freaking cool, like that. So it requires the hormone. Okay, so let's just draw this, and our book has drawings like this in the ninth chapter too. Let's just draw this like this, and say, we'll do it like this, where we say, we just talked about MyoD, and MyoD activating myogenin, and that MyoD binds the enhancer, and MyoD is a transcription factor that has to have two parts to the protein. One part binds to the DNA. So this is MyoD, and this transcription factor binds to the DNA, so it has to have a part of the protein that's called the DNA binding domain. And it binds to the specific enhancer sequence. You know, the letters are like A, C, C, T, G, A, like something like that. And this hand grabs it. This DNA binding domain is like a hand, and that's part of the protein. And then the other part of the protein here, that must be the way that it regulates transcription, right? We have to deliver it by having a DNA binding domain, and then we have to have this activity that we're delivering. And that's called the transacting domain. And remember, we're using the word trans like geneticist. It's from somewhere else. It's regulating this, but it's not in cis. It's not part of the gene. It's being delivered to the gene, and it's going to play a trans role. So it doesn't mean transcription activating, it means transacting. It's acting in trans, like that. And that's the thing that we now know, that's the thing that is bound to TBP. So that transacting domain has a shape to it that binds to TBP. And what must happen is that TBP binds, and at first it helps somehow get the DNA binding domain to be just the right shape to bind. Then it's there, and it's MyoD and TBP. And then it flops around, and suddenly it's delivered TBP to the PADA box in the promoter. And that was the rate limiting step, so boom, transcription start. Sweet, okay? All right, so that's classic transcription factorology. That's our title of our course, transcription factorology. So here's another gene. And there are lots of genes in this category. And these would be genes that are repressed. We'll see, they're turned off, not just off, but they're repressed in childhood, in both males and females. And then these genes get activated during puberty because there's high levels of estrogen bind to the estrogen receptor. So we're just going to call these estrogen target genes. And these would be genes that are activated in female vertebrates when estrogen is around. And there's lots of them, right? We've learned that transcription factors can regulate lots of genes. And there's a bunch of them. Yep.
Did you say it started on puberty?
Yeah. Yep. That would be our vision. And there's puberty for all vertebrates. All vertebrates, fish, have early growth period. They're juvenile. They're non-reproductive. They're like children, growing. And then they reach adolescence, puberty, and they mature to reproductive adults. All vertebrates do that. All of them like that. And so in our case, it'd be the female vertebrate. And we want to know right now that in female vertebrates in the juvenile, these target genes are actually being repressed. And then they get activated. Okay? So we're just going to call them estrogen target genes because they're really the target of estrogen. How does that happen? The answer is they all have an enhancer. And so here's an enhancer like that. And we'll now learn that people who study the estrogen receptor, they don't want to call that an enhancer because this thing is what's important for repressing the genes in, I'll just say, childhood, and then activating them. So that's a bad word. Enhancer sounds like activating, but it's both represses and activates it. So what these people love to call the enhancer, and we can learn this term just for this, is they like to call it a response element. And you won't hear that in other courses. You won't even hear it that much in this course. People like to just say enhancer. But when we get to estrogen receptor, we're like, hey, hold on. Because it both represses and activates. Like that. Yep?
Isn't there also things called like suppressors or repressors or something like that along with enhancers?
So we'll meet it. So transcription factors can either help activate or help repress. So help turn on a gene or turn off a gene, like that. And we'll meet a couple of examples of repressors. Most of the ones we'll learn about in our course are activators. So MyoD is an activator of target gene expression. This is both. And so people just like, I don't like the word enhancer. Okay? So I want to use response element. And responsive to what? So this thing is abbreviated an ERE. An estrogen response element. Because you could just take that piece of DNA and connect it to any gene, and the gene becomes estrogen responsive. Okay, all right? And so the transcription factor that binds here is estrogen receptor. So here's the estrogen receptor, and the estrogen receptor has a DNA binding domain. Every once in a while my pen just gets fuzzy, right? That makes sense. That's what's going to bind to the response element. So a DNA binding domain, then it's going to have a trans-acting domain. It's going to somehow regulate that. And this domain is going to be really interesting because sometimes it acts as a repressor and sometimes it acts as an activator. Right? Then, those are the two.
The trans works as a repressor and activator.
Yep. Yeah, somehow it does both. That's that. And then, right, MyoD just had the two domains. This has a third domain. So this has to have a domain that's called the ligand binding domain. That's where the estrogen hormone binds. Make this a little different color. So that's estrogen. Yep.
What's this thing at the very bottom?
Yep. So the estrogen receptor is an example of a transcription factor that has three domains. DNA binding domain that's got to bind to the response element. transacting domain, which is very interesting because it can be a repressor or an activator, and then a ligand binding domain. And the ligand binding domain is what is the switch. Okay, so I'm just going to redraw this so you can see that a little better. Right, so that's why I drew MyoD, is just to show us that MyoD has those two domains, and then estrogen receptor must have three. So we'll just draw this over with some thing here. And so here's an example of estrogen response gene, target gene. And it's the same one, and here's the ERE, the estrogen response element. And I'm just going to start over because I didn't really give myself good space, but it's the same thing. So estrogen response gene is the same one, and the example is going to be childhood. So we use humans as our example, but it's true for all vertebrates. Childhood, and then puberty, adolescence. All right, and so here's the estrogen receptor. The estrogen receptor is bound in the absence of estrogen in childhood, and at that time, the estrogen receptor transacting domain has a certain shape. So those are the three domains: DNA binding domain, transacting domain, and ligand binding domain. And then here's that same gene, that same receptor, estrogen receptor, during puberty. And now the estrogen is present. So there's a bunch of estrogen around, and it binds to the receptor like that. And when that happens, the transacting domain changes its shape. Like that. So we have those same things, see, and that's what happened. And so that's the dynamic switch in this case, is that the receptor shape changes because the hormone is there and binds, right? And let me just insert here. One thing you know is true about humans is that humans could choose to have hormone therapy to change their secondary sex characteristics. So a male human could take estrogen and start to appear to be more like a female. So the opposite is true. We're just focusing on estrogen receptor, but always the other thing is true about testosterone receptor. A female could take testosterone, start to be a male. So in this figure, we're going to continue to emphasize just in a female, childhood and adult. But I'm just going to add this here because it's just very interesting that this is the nature of our biology, right? Is that in childhood of a female or any time in a male. So childhood female or any time in a male.
What did you read in black?
Or male.
Oh, okay.
So adult male has the estrogen receptor bound to response elements of estrogen target genes, and it's repressing them. So that's just a little add-on based on the reality of our bodies, right? And types of therapy that can be done. So that's just a fact, right? Just an interesting fact that males can have some appearance changes by taking estrogen. That's how it happens. They already have the estrogen receptor, and of course they have the estrogen receptor target genes, They just don't have the estrogen. And so it's repressing those genes. And you can activate them. Yep.
You said estrogen response gene earlier, and now you're saying estrogen target gene. Are those like
Same thing. Yeah, sorry. Yeah. Sorry. Okay. Yeah, sorry about that. Thank you. So,
What is the transactional domain for plasma?
what's the transacting domain doing?
Yeah.
Yeah. So let's talk about that next. Thank you, Godwin. Estrogen response gene or target gene. Yeah. Those aren't, really technical words. They're just trying to describe a bunch of like that. Okay. So what's happening here? Okay. So what's happening is that in the child female or the male, anytime, the transacting domain has a shape that causes another protein to bind. And this protein is an enzyme, so that's what I've drawn here. So it's recruiting an enzyme to this. Remember, it's tethered, and so it's just recruiting an enzyme to this area. This enzyme, like a bitey dog, it's going to catalyze some reaction at a high rate because it's stuck there, like that, okay? And so this is a transcription factor, but notice that it is not bound to the DNA directly. So this transcription factor doesn't have a DNA binding domain. And transcription factors like that are called co-regulators. So the general term for this is a transcription factor that's a co-regulator, and it is got to bind to another transcription factor that is bound to the DNA. And this one happens to be co-repressor. So it's a co-regulator that's a repressor. Wow, repressor. No, it's like that. And then we're not going to talk about its actual what is that doing, but what it's doing in general we'll learn about is it's remodeling the chromatin. And what it's doing is it's folding the gene up so it's not accessible. And we're just going to name the enzyme activity now, and then we'll learn later when we talk about chromatin. Oh, okay, what's this? Okay, so the actual enzyme activity is, I shouldn't write it right here. It's called a histone deacetylase. And so we'll just learn that term now, but we'll learn later. We'll fit it in, so we'll be like, aha, there's a histone and there's a histone deacetylase. That's its enzyme activity. So it's doing some enzyme activity called histone deacetylase, like that. All right, so then notice that when we add the hormone, the shape changes of the receptor. So down here, when the hormone's present, the shape changes, and it lets go of that histone deacetylase activity. So in a female child, or a male at any age, there's a histone deacetylase that is doing enzyme activity that causes these estrogen response genes to be folded away and not used physically, okay? And then what's happening here? The shape changes, so it's not holding on to that, but now it's holding on to a different co-regulator, which is also an enzyme. So it lets go of the histone deacetylase and it grabs a different one. It's also an enzyme. And so this is a co-regulator, transcription factor, and this one would be called a coactivator. And it has an enzymatic activity that is exactly the opposite. So this is called a histone acetylase.
So does a co-regulator not bind to the DNA, but co-activator binds to the DNA?
No, co-regulators do not bind, and we have co-regulators, some of which are co-repressors and some of which are co-activators. So this is
Right.
so both these are co-regulators.
Gotcha.
They're transcription factors that are helping regulate, but they're called co because they need to help somebody that's bound
Gotcha.
to the DNA. Yep.
It's like I understand like these facilities and facilities, these things are, but can you just describe like, are they like what it's turning into? Like what the estrogen response gene is turning into, is that what you're saying, or are they like?
So just to describe that, the histone deacetylase enzymatic activity causes the estrogen response genes to get folded up like a blanket that you put away for the summer.
Yeah, I'm just
And-
asking, is it
yep.
like, is it the whole complex as a whole? Is that what it's, like after everything binds together, is that whole complex the histone, what, you know, the activator or the deactivator one?
I don't know not sure what you're asking, but remember that we're just naming the enzyme activity right now.
So the enzyme activity is called the histone,
Huh?
it's just an activity that's happening. It's not
Yes,
like an actual
yeah,
protein.
like enzymes catalyze a certain chemical reaction. What is the chemical reaction? It's catalyzing histone deacetylase. So maybe to just mention a little bit more, so if you remember your chemistry, there is a chemical group called an acetyl group. And we'll learn there are some proteins that are part of folding the DNA. They're called histones. And so the histone acetylase activity adds the acetyl group, chemical reaction, covalent bond, adds the acetyl group, and that causes the chromatin to unfold and allows the gene to be expressed. It's the rate limiting step.
And that's what estrogen is doing.
Yep, estrogen is unfolding the blanket for the winter, physically. And the way it does that is by recruiting this histone acetylase that acetylates these histones, and they just open up like that. And then the histone deacetylase activity removes acetyl groups from histones, and that just causes the DNA and the chromatin to fold out. And we'll talk in more detail about, well, what does that mean? But yes.
Can you also like discuss something about like methylation as well?
Yeah, so methylation. So we've already talked a little bit about DNA methylation, and that's different than this, right? We're not talking about methylation or DNA. We're talking about these histone proteins and acetylation. And you'll see that these are examples of chromatin remodeling. So remember, that's what we're talking about. And of those, there are many different types. And so we'll learn a little bit. Our book talks a lot about this, but we'll try to drive through with some specific examples. And one of them is going to be histone acetylation, like that. Another one that we'll learn a little bit more about is DNA methylation, and that's different. There are some things that are confusing, when you start adding things. So I'll just say, for example, histones can get methylated. Darn it, that starts getting confusing. So we'll try hard to go through on just a couple of examples that we'll become experts on and be able to write about on the exam, and then read about the other ones, but recognize that we don't want to get confused by too much information. Yep.
Can we go back to the drawing?
Absolutely, because this is just nuts. This story is nuts, because are we talking about our bodies? Oh yeah.
So
Like all vertebrates that have ever lived. Did Tyrannosaurus Rex walk around with this situation? Yep.
I just wanted to make sure the histone acetylase and the histone deacetylase is the physical change to zone 2 on the ERE.
Yes.
Okay.
So yes. So
And then
the green is the estrogen receptor protein,
Yep.
and the 2 is its transactin domain. That's the part that's going to do the job of regulating transcription, and all it does is it changes shape. When estrogen binds, it's one shape. When it's not bound, it's another shape. And it's a binding site to just tether these enzymatic activities, which are opposite.
So does the histone deacetylase and the histone acetylase refer to that, or are they referring to the protein that binds to the zone 2?
Yes. So the enzymatic activities are naming the protein
Gotcha.
co-regulator. So that blue protein is an enzyme that is a histone deacetylase enzyme. That's its catalytic activity. That's its function, is to remove acetyl from histone. It's a co-regulator. It's a transcription factor that needs help to bind and get located, and it's a co-repressor, like that.
Yep.
So all those blue words apply to that blue protein.
Yep.
And this is great because this is super physical, right? And you're seeing that, and I'm trying to, it's this, our example.
Yep.
And we're introducing that example, then we're going to say, and we're going to talk generally about chromatin remodeling and these changes like that. And as we do it, we're having estrogen receptor in our mind going through, because we're going to be experts like that. It's a lot like how we used muscle differentiation at the start of our course as an example of changing gene expression. We're using this as an example of a dynamic transcription factor, ligand dependent, and then one that regulates target gene response genes by changing the chromatin. And it does that by recruiting enzyme that causes the gene to be folded up and inaccessible. That's the histone deacetylase, or open and accessible, like that. Yep. Yeah. So I could have made a video, right? A video like that clay model video, and just been like, this is a really satisfying thing to learn about, because there it is. It's our specific example, super significant and exciting example, but also lets us have something in our head to go through the general topic of chromatin and chromatin remodeling. Other questions people have right now? Let me just, as we go into this chromatin, say that, again, chromatin is the DNA plus proteins that are bound to it. So in our clay model videos, we just took a piece of clay and said, that's DNA. But in reality, in the cell, the DNA is always bound by a bunch of proteins, including these histones that we'll learn some about. So that's chromatin. And chromatin remodeling means to reorganize that. And this is a good example of either folding it up or unfolding it. So let's just add to one more thing to this, just to introducing these things. And then we'll go to a little concept chat action. So when the chromatin is folded up, like the blanket that's folded up for the summer, that's called heterochromatin. Heterochromatin, one word. And I know this is getting messy, but that's called heterochromatin, or another fine name for that is, it's also called closed chromatin. Isn't that a great, happy name? Closed chromatin, inaccessible, off. And then down here, this is called euchromatin. Or the other synonym is open chromatin, and that's a nice word. So we want to know those words. And specific example, but when genes are being expressed, transcribed, they are in a euchromatin organization, and then when they're not, they typically are in heterochromatin. So you can kind of think of that as kind of transcriptionally off in the top or on below. Yes, Hannah?
Isn't there like two forms of like heterochromatin and chromatin? There's like the ones that are like always permanently inactive facultative and
Yeah.
constitutive.
Yes.
Is that
Yep.
chromatin?
Yep, that's heterochromatin. And we'll just briefly add that. Can you just say, did you learn that in what?
Genetics.
Genetics. So like a Ken Weber thing?
Yep.
Yeah. So just to mention that, so this heterochromatin is all folded away and condensed and closed like that. And this is an example of heterochromatin that could get unfolded.
So I'd say it's, from what I remember, faculty.
Yeah, but we're not going to learn that term. And just to answer the question, so there are genes that could be folded away or get unfolded. And that's one kind of chromatin, and I'll just say facultative. And then there's another kind that's found like at the centromere, so there are structures of the chromosome where it's like going to bind to the kinetochore, or at the telomeres, and those are called constitutive heterochromatin. They're just always folded away like that. But we're not going to learn that term because we really like genes, and those regions of constitutive heterochromatin really have few or no genes because they're more of a chromosome structure. Yes. So, but yes. For us, heterochromatin is just heterochromatin of genes. They're folded away, transcriptionally inactive. They could get unfolded into open chromatin, euchromatin, and transcription. So that's what the estrogen receptor is up to. That's its function. Yes, it's a ligand-dependent chromatin remodeler. And it's so interesting that it could be a repressor or an activator. So would we want to call the estrogen response element an enhancer? No, it's just not a good word. We need a more general word, so it's called an ERE, an estrogen response element, like that. Okay, all right, let us do, let's see, did I get the sign-up sheet handed around? Yep, okay, so the sign-up sheet's floating around, and we will do a concept chat. So say hi to somebody that you don't know, because we still haven't met everybody in class yet. And just take a couple of minutes to talk about these 3 things that you think we talked about today that are important. Sure, yep. Yeah, and you can sign in. Do you go by Brooklyn or Brooke? Brooklyn? Brooklyn? Okay, Brooklyn.
Yeah, I had a flat tire this morning.
Oh, I had a flat tire last night. Or yesterday morning, actually.
I think I ran over a nail.
I ran over a screw, but they were able to fix it, they think.
Yeah, I had trouble like they came and fixed it for me.
No. Anyway, all right, welcome.
They have like a DNA binding versus like enzymes that don't, so they have to come together to facilitate
just went more in depth on the ways that the ways that transcription factors regulate
TDP bind
It's if we didn't get just copy the first page and we have to so my idea was where else
What did you say?
some of the Preinitiation complex.
What did you say?
I bring them
Yeah, it's the 2 steps.
oh 2 steps of Preinitiation. I didn't write them down. So it binds the tyrosine and remodeler. And that was like the when they said
When we're talking about these important things, we're talking about possible ideas for tattoos. And so I'm just looking if there's anybody that has a tau-binding protein tattoo. Tau-binding protein is a pretty famous protein, but I don't see. Oh, that's a pretty good one right there.
No, I'm just wondering this thing in general.
Okay,
It's a protein,
looks
but
like
it's a transcription factor.
that's a good tattoo.
Yeah, it's a transcription, but it's also a transcription factor.
yeah, so we've got the DNA.
No, but it binds to
Doodle-doo.
Myod isn't a transcription factor, is it?
Myod is a transcription factor.
Okay.
Yep.
Sorry.
So so this right here, the complex that we made with the Myod transcription factor and how it's a protein and how it has TBP binding to it, is that this like element, this thing here, this protein, the Myod transcription factor, is that a receptor that binds to the DNA, or is it not a receptor because there's
No.
nothing binding to it? It doesn't have any ligand thing.
That's a good question, and we'll talk about that after this concept chat. Yeah, we'll answer that question.
Because I just switched to the fat one, so then you're drawing.
and remind me if needed. Yeah, we'll talk about this for another minute.
This thing here
So
has
there's
the
a
same,
book how we talked about the
essentially the
receptor
transcription
factors or activation
this one,
depending on whether
but
or
this
not
one has like this thing here, it's considered the estrogen receptor. That's why I believe this is considered receptor. I don't think it is.
Because
It's
that's
about that one.
That was just
Um,
an active
so
role on your team, you're right.
Yeah.
This is the
So like
receptor.
when you have puberty.
Okay,
Because it's-
so the, but this whole, is this not whole thing not considered estrogen receptor, or is it?
Well, that's the, yeah.
It
it's
really
the
threw me
estrogen
off at first that the receptor was
Transcription.
binding to the DNA.
Estrogen receptor, transcription.
But I was
Well,
thinking
it's
receptors
the
was like all
estrogen
cell
response.
surface. And then I was like,
Okay,
oh,
estrogen
it's a receptor.
Instead of a
So-
promoter,
Binding.
instead of this being the promoter, it's an estrogen
Yeah, because I didn't
response.
say that it's called,
So this whole thing,
I was,
this whole
I was
like-
thinking of the
receptor
that
and
it's called
changes to
response
be a
element
response element.
Yeah.
with that.
Because it's not, it can either be yes or no, and an enhancer means
In this situation, it's
yes
not
or
necessarily
active,
like active.
but if there, it sounds like it, then it's a no.
So like
Okay, TIBA. What is one thing that you guys talked about that you thought was important today, or like worth a tattoo today?
Yeah, we talked about how myogenic transcription factor has two domains, and one of the first one binds to the DNA binding domain, and the other one, the transacting domain binds to the TBP, and how the estrogen target genes has three domain actually, the ligand, the DNA binding domain, and the transacting domain.
Yeah. Yeah. Right. And so, we started our course with our clay model and we named MyoD. MyoD is a transcription factor, and we have our clay model understanding of that transcription factor. And what we did today is we just added a little bit of information to it. That it's got two parts, that makes sense. The DNA binding part has to bind to the enhancer, and then it has to have this transacting domain that is somehow going to increase the transcription of the myogenic gene. And it does that by interacting with this TBP. The rate limiting step is TBP is not there, and it's part of the preinitiation complex. So it's a sweet example. And then it just got a little bit complicated because it's like, whoa, TBP is helping MyoD first, get to the enhancer, and then they're there, and now the MyoD is tethered and can help flop the TBP into place, right? Like that. Okay. Great. Then let's see. Jeremy, what's a thing you guys talked about?
We talked about ERE, the estrogen response element, and that it can be in two phases, the heterochromatin and the euchromatin, depending on the presence or absence of a ligand.
Yep. Yeah. And so there, right, the estrogen response element is an enhancer. We just want to use a better name for it because the estrogen receptor binds there, and the estrogen receptor could be a repressor in the absence of the hormone estrogen ligand, or an activator in the presence of the estrogen hormone ligand, right? And then that becomes a really beautiful example for our list of transcription factors and how they get switched. That's one is a ligand-dependent, hormone-dependent transcription factor, and it switches. It's a little molecular switch, like a little light switch, and it's like, it's off, childhood. It switches on in females, like that. Yeah, fantastic. And then, Zofia, what's another thing that your group talked about?
Well, we also talked about the steps of MyoD, like the two steps. We also talked about how all of coding genes are transcribed by RNA polymerase II.
Yep. Yeah, and that's just a little piece, right? We skipped chapter 8, and one of the pieces in chapter 8 is it describes, people organisms like people have 3 RNA polymerases. Okay, fine. We're talking about RNA polymerase II. The RNA polymerase II genes are organized like the ones we're talking about. The ones that are regulated by RNA polymerase I and 3 have a slight different emphasis like that, but it's always 2 parts, the transcription unit and the regulatory region. Yes. Okay? And then Hannah asked a question, so let me just clarify this. So we got a few more minutes to talk, and so let me just talk about this. So you remember that I said, oh, you know, this clay model muscle differentiation video, there are these two cells, and we're talking about a gene, and then we're talking about a transcription factor. And we could just have, like, we fill in the blanks, like, what are the cells we talking about? What are the context? And then in them, what gene are we wondering about? And then what is that transcription factor that's the really main one regulating that gene? So we kind of have a fill in the blank like that, because it's just a universal story like that. So when we get to chapter 17, that's also a universal story where we fill in the blanks. And so in chapter 17, we'll see, here's a cell, and we'll fill in the blanks of, there are some receptors. And one class of receptor is going to be the receptor tyrosine kinase receptors, like PDGF and insulin receptor, and we'll learn about KIT, like that, you know? There are 58 of them, 60 of them in humans. So that's one category. And then we learned, oh, RAS is activating it, like RAS and that pathway. But in chapter 17, we'll learn about two other pathways that could be signal transduction pathways activated by these guys. But that's just one category, fill in the blank. And then in chapter 17, we'll learn about another really famous type of receptor called a G protein coupled receptor, of which there are hundreds in our bodies. Like it said, 70% of all drugs, all pharmaceuticals, regulate G protein coupled receptor pathways. Like that's how all those drugs work, like that. And we'll just talk a little bit about like that. Then there are maybe 3 other specific receptors that don't fit into this, but the book talks about, maybe it's 4, that we will want to learn about a little bit, like that. So they look like that. And these are all receptors. And they're all in the cell membrane. And one of the things we learn a lot about in our course is that these receptors will have signal transduction pathways that are going to affect a transcription factor. Like here's one, PDGF binds the PDGF receptor, activates RAS, and that activates ALK, a transcription factor. So see how the receptors are in the cell membrane, and a lot of times we're interested in how they're affecting the cell, and it includes changing gene expression by regulating a transcription factor. All right? So I think what Hannah was a little bit confused about, it's good to draw this, is that the estrogen receptor is inside the cell. And it's a transcription factor. So it's in this category. And what's going on here is that estrogen it's very hydrophobic, and it can just diffuse through the membrane and bind to the receptor inside the cell. So we have the estrogen receptor, that's estrogen, and the estrogen receptor that we're talking about today. And so there's a receptor that's a curious one that's actually inside the cell, and it's a transcription factor. So we have to be careful to appreciate, you know, all these other receptors are cell membrane receptors, and their ligands are outside the cell, and they have to regulate signal transduction pathways that could include changing transcription factor. And so see, this is another drawing we could do, you know, and it's a universal drawing, fill in the blank. So that's the estrogen receptor. And in us, there are some other receptors like this. One of them is the testosterone receptor, like that. It turns out that this is a class of receptors in us, and they have a really nice name. They're called nuclear receptors because they work in the nucleus. And for all of them, their ligands can be inside the cell. They can float through the cell membrane, or they're made inside the cell, like that. So these are nuclear receptors. In humans, there are 48 nuclear receptors, like that. And we're focused on one. and they're in a family, a group, and they are really important in our health like that, okay? So, to see that a little bit, like, because Hannah was asking transcription factor versus receptor, right? And we want to be careful, you know, for most of our course, the receptors are in the cell membrane, the transcription factors are in the nucleus. And here we have this curious one that's an example of 48 in this category of a nuclear receptor. And nuclear receptors are really cool. Estrogen receptor is really cool because it's a transcription factor and the ligand is switching it on and off. Yep?
But doesn't what I'm confused about is the estrogen element has a receptor on it that takes the binding, or is it that the ligand for the estrogen to actually bind to?
Yeah, so
So in
it's,
comparison to the MyoD that doesn't have that receptor on it, it just has the DNA binding domain and the trans something domain. So that's all it is.
yeah, yeah. And so what we have is a transcription factor in the estrogen receptor that's a little bit fancy because the hormone is the switch. And it has to have a ligand binding domain to bind that hormone. And then the entire shape of the receptor changes some.
You mean the whole
The estrogen receptor.
but I thought the other thing that changed was the cysteine as well.
The cysteine?
No, so you know how you have the estrogen, what's it called again, the estrogen?
Response element?
Yeah, response element.
Yeah?
We have those two things. We have one shape when it's not bind to.
Yeah, the transacting domain, yeah.
And then when estrogen binds, it shifts it, and then it changes that domain, and then it can fold over and actually activate it. So that's all. I think that's all. Is that correct? What
Yep.
you said? Okay,
Yep.
that's true.
Yep. Yeah. Okay, and we're way past stopping time, so we got to stop and get out of here. Yes, and but you can see that